Hey everyone, my name is Elora Lopez-Nandam, and today I'm really excited to tell you about kinship and selection in aquarium-bred Acropora hyacinthus corals here at California Academy of Sciences. Rebecca Albright and her team started the coral spawning lab in San Francisco a few years ago, and in 2019 and 2020, we had two really successful coral spawns. What you're seeing here are corals that were collected in Palau, brought to San Francisco, where they spawned in captivity, and then researchers took that spawn, fertilized the gametes, produced embryos that developed into larvae, and then finally into settled polyps. And now this is what it looked like 2 years later. These are 2-year-old corals that had been born in the lab and were thriving and doing great. And now we're in December, so they're 2.5 years old and looking really good. But when I joined Cal Academy in the Albright lab in the spring, my question was, how do we combine coral husbandry and genomics to start to inform coral conservation? And so in spring of 2021, I sequenced the genomes of the 3 coral colonies that spawned in 2019 and the 13 coral colonies that spawned in 2020 that had all been collected from the wild in Palau and brought into captivity. We also sequenced the genomes of the 9 babies that were born in 2020 that survived to be at least 1 year old. In 2021, they were a year old. And the 23 babies born in 2019 that at the time were 2 years old. And my objectives in doing this was, one, to create pedigrees and family trees and know how all of our data or all of our animals in this system were related to each other. And also to start to try to figure out if we can identify signals of selection induced by the lab environment, simply because the lab environment is so incredibly different from what baby corals would be experiencing on the wild, on the reef. So very briefly, we extracted DNA, prepped full genome libraries, mapped all of our sequences to the new chromosome level Acropora hyacinthus reference assembly that we put online earlier this year, and then called high quality SNPs that were genotyped across all of the individuals in our data set. So a SNP, or a single nucleotide polymorphism, is a really common form of genetic variance in a population. So what we're saying when we say SNP is really, if we're looking at a sequence of DNA in one individual, there's one nucleotide that's different than in a second individual. In our data set, I identified 16.9 million SNPs that passed all of our quality filters for every one of our 47 individuals. And so all of the results that I'm going to show you today are based on these 16.9 million SNPs. So the first thing to do was assess identity by descent to start to reveal kinship. This was done in a program called PLINK, and it calculates pi-hat scores. And I'm not going to get into the math here today, other than to give you this cheat sheet and tell you that if 2 individuals, when compared to each other, have a score of about 1, then we would say that those are clones of each other. If it's about 0.5, then those are likely full siblings or a parent and offspring. And if it's about 0.25, then these are half siblings. So what I'm about to show you is going to be a heat map in which we look at pairs of individuals across the data set. So if you're watching a recording of this, I encourage you to pause for a moment and start to look at the pi-hat scores between different individuals. If you're not watching the recording, that's okay. Here's a much simpler way to start to visualize how all of these individuals were related to each other. So for the 2019 individuals, we found that 22 out of the 23 offspring that survived to be 2 years old were all full siblings of each other. They all shared these same 2 parents. And just one of them shared or had 2 different parents. So that's really intriguing. And we find something somewhat similar for the 2020 data, in which the majority of colonies that spawned in 2020 had no offspring that survived to be 1 year old. 3 of the colonies produced 6 of the offspring. So we have 4 siblings here and 2 half siblings. So we have kind of a coral love triangle going on here. And then an additional coral baby at 1 year old from 2 different parents. So from this data, differential parentage is really clear. And my question was, well, how much of this can be explained by selection. How much of this is because certain parents produced offspring that were more fit to survive in the lab environment than others. So the way that we start to look for this is to identify SNPs that are really different frequencies in the surviving aquarium-bred offspring as compared to the wild-caught population from Palau. And since we did this twice in both 2019 and 2020, we can start to use the 2019 and 2020 cohorts as replicates. So the data that I'm going to show you today is very methods light. So if you have questions about how we actually went about doing this, please ask me later because we're a little pressed for time. But as I mentioned before, we started with 16.9 million SNPs. And on the x-axis here, we have the F1s, so that's the offspring allele frequency at each SNP, minus the F0s or the spawners allele frequency at each SNP for the 2019 cohort. On the y-axis, we have the exact same thing, but for the 2020 cohort. So if you see a really high value, then that means there is a pretty large difference in allele frequency for the F1 versus F0 populations for a given cohort. So the vast majority of these SNPs are what we call background SNPs, or they're quite close to zero. So there's not that much difference in allele frequency between the F0s and the F1s. So if we eliminate all of the background SNPs, then we come down to only 250,000 SNPs. But again, as I mentioned before, what I'm really interested in here are things that were shared outliers for both the 2019 and 2020 cohorts, not those that were just in one or the other, because that indicates that it was more stochastic and not particularly relevant to selection. So if we eliminate those, we come down to just 1687 SNPs. And if we only look at those where allele frequency changed in the same direction in both 2019 and 2020, then we come down to 652 SNPs. So what does that mean? Well, first of all, the fact that we went from 16.9 million to only 652 SNPs indicates that in order to find signals that are very rare like this, you need to start with a very large data set. But also, we know that 41 of these SNPs are encoding regions, and 173 of these SNPs are upstream of genes. So we're starting to look into which genes these affect and what it might mean for coral breeding biology. And I'd like to thank everyone who made this possible.